The insertion or deletion of a single nucleotide can have a significant effect on the protein produced in translation. This is because ribosomes depend on a specific reading frame to correctly identify which codon it is reading. The reading frame is dependent on the start codon AUG, but an insertion or deletion after this point can result in a shortened or elongated protein. Imagine shifting each letter in a sentence down by one letter or up by one letter. It makes the whole sentence nonsense, and just like a protein formed without the correct reading frame, it is non-functional. Let's take a look at the protein sequence: methionine, tryptophan, phenylalanine, threonine, lysine, glycine, and so on. Now we know that some mutation involving a single base pair results in the protein sequence methionine, tryptophan, isoleucine, and histidine, but we want to know exactly what change in the mRNA caused this. In this case, the insertion or deletion resulted in a truncated protein which will not function properly. Let's start by looking at all the possible codons for each amino acid in both the normal and mutant proteins, which I am writing down. Methionine and tryptophan only have one codon apiece, so we know which codons they must have. The first actual change is between the phenylalanine of the normal strand, and it becomes an isoleucine in the mutant strand. What change would make a phenylalanine codon code for isoleucine? I think inserting an A in the first position would, but now we have to figure out which isoleucine codon is actually being used. The AUU codon follows the theory of an A being inserted, but also ends with the first two letters of every single phenylalanine codon. And now to figure out which phenylalanine codon is used. So which codon for phenylalanine also has the first letter of the histidine codon? UUC. And I'll just follow this process all the way down the chart. Notice I also included the stop codon to ensure that my mRNA sequence is complete. Now that we're done, my favorite way to check my answer is to write out both the normal and mutant sequences and line up the bases of the two strands, ensuring that each has a match except the A that was inserted. And this is how you can figure out the mRNA sequence of a mutant protein strand.